Yes, moving on with the muscle contraction. As we are seeing the thin filament side, first of all, we have completed the thin filament side, and now we are moving with the thick filament side. The thin filament side, I'll remind you that what happened, the thin filament is of three: actin, proponin, and tropomyosin. Actin, proponin, and tropomyosin are present there. And thick filament side, thin filament side, what is the requirement? Exposure of active site, which is actually active sites are located on F actin. They are covered by tropomyosin and supported by TNT and TNI of troponin. Now, what happens that the unlock is done by calcium ions. Calcium ion will come and bind with the TNC. Calcium ion will come and bind with the TNC. That calcium troponin complex lift the troponin tropomyosin complex. See the complexity. The calcium troponin complex lift the troponin tropomyosin complex that make the exposure of active sites. So active sites are ready now. Now need to go with the myosin, the thick filament side. Each myosin is with that like three portions in each myosin: head, neck, and tail. The two tails of two myosin molecules are coiled like this. On above the myosin, we'll find two sites. One is the actin binding site, which binds with the active sites of the actin, and other is the ATP binding site. Other is ATP binding site. Okay. So actin binding site we are talking about. This is the actin binding site, which binds with the actin at active site. In this place, we have to talk about this particular place. Okay. Then the other is ATP binding site. That means the ATP molecule will be connected here. So ATP will break down in the form of ADP plus phosphate ion. This particular site we see. Okay, so what is the requirement this side? This particular area is having already ATP. The myosin head is having ATP binding site. Do ATP gives energy? No. This is very important. In the ATP form is the storage form. So what is required? Breakdown of ATP into ADP plus phosphate ions is required. For that, what is required? There is an enzyme called ATPase enzyme. That means for the activation of the myosin head, what is required? Myosin head activation required ATPase enzyme. Just see the difference. What is the requirement for the activation of this particular exposure of active site? Calcium ions. When calcium ion comes, the exposure of active sites are happening. Now, we said, what is the requirement this side? ATPase enzyme. Who will bring ATPase enzyme? Calcium ions will bring. That's all. The calcium ions will bring. So that means the calcium is the hero for muscle contraction for both thick filament side and thin filament side. Okay. So just combining these two will make the muscle contraction, and we need to see how the calcium ions came. Okay. So just let me show this particular in the diagram. Let me show that. Let me clear the ink here. Then this area, we just talked about this area is actin in the form of F actin like this. This is the F actin. Okay. And talking about the myosin, myosin is with head. And these kind of questions nowadays we are not getting. This is not the MSET. That in MSET we'll have some depth questions like this. The head is towards jet line, then tail is always towards the head zone. These kind of questions we used to get in those days, but not now. That basic concept based questions are only going to be asked. If you remember, no matter. The myosin head is always towards the jet line, and the particular tails are always towards the head zone. So, with the help of this particular head, the myosin head that is going to pull the thin filaments. Let me show this particular in the zoom. So, this is the myosin head. The myosin head is going to pull the thin filaments, attaches the thin filaments at active site and going to pull that particular thin filament towards the head zone. This is the story. Okay. So for that, what I said, you need to remember the thin filament side, how does the activation and exposure of active sites are happen? And in the thick filament side, we need to remember that how does that particular thick filaments myosin heads are getting activated. In the both cases, we said about the story of the calcium ion. So now the question raises here, how does that particular calcium ions are getting liberated? How does the calcium ions are getting liberated? This is the sarcolemma. So I'm not going to draw the total here. The first of all, the pepimycium, then after perimycium, then after endomycium. Finally, we reach to the sarcolemma. Okay. From the central nervous system, from the brain, the nerve fibers get started, crossed the epimycium, perimycium, endomycium, and now they are having synapse with sarcolemma. Now they are going to make the synapse with sarcolemma. So, where does the muscle contraction is going to happen? In the muscle fiber. This is the muscle fiber. Let me write here for our remembrance. This is the muscle fiber. So each muscle fiber is having many myofibrils, isn't it? That each muscle fiber is with many myofibrils. So what do you see here? That let me draw all Z lines here. Only Z lines I'm going to draw. Sarcomere, sarcomere, sarcomere like this. Okay, so Z lines, Z line, Z line. So these are all sarcomeres. Make it, these are all sarcomeres. Okay. Then, okay, we'll make it H zone also. It's very difficult here to do like this. Mm. So there are so many sarcomeres. Now our nerve fiber came and synapse with this particular sarcolemma. We call this particular area as motor end plate. This is said to be as motor end plate. The acetylcholine neurochemical substance is getting released. Who is the postsynaptic membrane in this case? Sarcolemma. So sarcolemma is getting depolarized now. Information came from the brain. We are talking skeletal muscle. That's why we say the information came from the brain, and the acetylcholine is getting liberated, and sarcolemma is getting depolarized. The depolarized sarcolemma invaginate inside. That is very important. Invaginate inside. So where it is invaginated is going to be matter here. So in case of that other mammals, that is the invagination through the jet lines that I shown now. That in case of human being, we'll find that invaginations in between the AI bands like this. AI bands. Okay, so this is not much required. Actually, we are having this particular information in the state textbook in detail. Whatever invagination you need to recognize first of all. Okay, so transverse tubule we can say. What is transverse tubule? Invaginated sarcolemma is called transverse tubule. The either sides of this particular sarcolemma, we can see the cisternae of sarcoplasmic reticulum. Let us use another color for this. Yeah, that's very new, brand new cisternae of sarcoplasmic reticulum. Okay, so let me see this. This is transverse tubule, and these are the cisternae of sarcoplasmic reticulum. This is called triad system. What is triad system? That invaginated sarcolemma in the form of transverse tubule, either sides of that particular that T tubule will find the cisternae of sarcoplasmic reticulum. The cisternae of sarcoplasmic reticulum is called as the triad system. Now let's make the sequence here. Let's make the sequence that from the neuron, that axon terminals, where the motor end plate is the sarcolemma here, both synaptic membrane, which is getting depolarized. From there, it will go to the transverse tubule. From the transverse tubule, on either side, will find the cisternae of sarcoplasmic reticulum. These cisternae of sarcoplasmic reticulum will collect the information from the tubule, depolarization information. They release calcium ion. So our hero released calcium ions. Who release the calcium ion? Cisternae of sarcoplasmic reticulum. What the calcium ions will do? That we have. Calcium ion will bring the ATPase enzyme and breaks the ATP and activates myosin heads to pull the thin filament. What about thin filament site? Calcium ion will bind with the TNC and form calcium troponin complex that lift the troponin tropomyosin complex for the exposure of active site. Everything is ready. Now let us try to see that particular video. Before that, let me show you that the muscle contraction procedure, the procedure of muscle contraction. Wait. That means synapse procedure we are talking about.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium.
Wait. Yes, I want to show you that particular the video. Wait, it's coming. The muscle contraction video.
An action potential arrives at the presynaptic.
Did you get the video? Yes.
Synaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholinesterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
Yes, then
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic.
yeah, let me continue. Then what I'm saying here, when you start running, just we'll take a smart way here. The 2 minutes will take here to understand this much better. When you start running, so what you use first, we use stored ATP first of all. Okay, that stored ATP is over. Then what we do? We have muscle phosphagen, creatine phosphate. Creatine phosphate provide phosphate to the ADP to make it ATP. Okay, that is the second step. All the phosphate is over. What we do? Breathing started. That means more oxygen we use it to take to break the more glucose molecules. Okay, that is a step 3. Then you are running, your skeletal muscles are much faster, they are moving. That oxygen depth will come. What is oxygen depth? Your supply is not equals the demand. More oxygen they require more energy, but your oxygen is not sufficient. What do you call oxygen depth? Now they depend upon that particular anaerobic respiration. Without oxygen, what we can do? We can do glycolysis. Right? Nothing but called anaerobic respiration. Okay. So that anaerobic respiration can be done. That anaerobic respiration makes over the production of lactic acid. That pyruvic acid will be converted into lactic acid. Okay. This lactic acid makes the pain. That means when you start running, when you are continuously running, what's happening? The steps and events we are going for. We use ATP first. We use creatine phosphate to provide phosphate to the ADP. We use oxygen. Then we go for anaerobic. When the lactic acid is formed, that means we can understand the pain gets started, and we are undergoing, we are ready to go for fatigue. Okay. So the particular fourth step, last step is going to be fatigue, anaerobic process. When you take rest, what happens? That muscles lactic acid from the muscles will come to the liver through blood circulation. There it will be converted into glycogen, and we send glycogen back again to the muscle. That's why when after running you feel pain. Why? Lactic acid. When you sit for a period of time, you feel relief because lactic acid from the bloodstream reaches to the liver will be converted back again to the glycogen. So this is a story of muscle contraction, and with the skeletal system will continue tomorrow, as we are taken much time here because the more applications we have in this particular area, so we need to focus in this area. Okay, thank you.